This lecture is going to take us through the genetic diseases. I have listed here chapters 5 and 6. 5 is basically a review of concepts you should already know, so I included it only to say this is something you may need to go back and take a look at. For the most part, there won't be anything on chapter 5 that shows up specifically on exams, but there is an expectation that you understand those concepts so that you can look at the genetic diseases that we talk about in chapter 6. So when I'm talking about those things that are things you should already know and reviewing, this is what I'm talking about here. You should know what DNA is, the deoxyribonucleic acid. It's the hereditary material in all of our cells in the humans, and actually there's DNA in all sorts of organisms. Anything that has a genetic code is DNA in some form or another. You should know what RNA is. There are multiple types of RNA. Messenger RNA makes the copies of the sequence from the DNA and carries it out to the ribosomes to make the proteins. You have ribosomal RNA, which is what is responsible for making up the ribosomes. And then transfer RNA is involved in translation. And there are actually several others as well. So you need to have a basic understanding of that. We talk about the nucleotides and replication. Nucleotides are the components of the DNA and RNA, and the replication refers to either the copy of the DNA itself when cells are dividing, or actually the copy that's being made to the RNA. So that term can go for either process. Protein synthesis is really the whole point of the DNA code. It makes proteins. That's what DNA does, is it codes for proteins. And so it's broken into segments that are referred to as chromosomes. So in the nucleus of each cell, the DNA molecule is packaged into these thread-like structures that are called chromosomes. And each chromosome is made up of tightly coiled DNA that's wrapped around proteins called histones that support it. You can't see it till the cell divides, and then when it divides, it condenses into chromatin. Genes are on the chromosomes, and the genes are pieces or sequences of DNA that actually are responsible for this protein synthesis. They code for different proteins. So we have somewhere between 20 and 40,000 genes. There's a lot of arguments among researchers still as to what exact number of the genes are actually viable. We have it mapped, but we don't really understand everything about it yet. But we're getting better. Every year I teach this, there's new articles and new things out there about the genetics. What we're really looking at now, though, is going forward into chapter 6 is the ideas of gene expression and gene mutations that lead to disease. And let me just say right here at this point, there's a difference between what we say of disease is genetic versus a gene is hereditary. I will argue that many, if not all, diseases in some way interfere with that process of protein synthesis and what's happening with our genes. So all disease processes in some form or another has a genetic component because all we are is composed of the messages of our genes. But when we say something's been inherited, then we're saying that particular gene is being passed from parent to child. That's heredity, and that doesn't necessarily make it a genetic disease. We use that term way too loosely in society, in science. All diseases in some way or another are involved in something with our genes, with our genetic makeup, but not necessarily something we pass from parent to child. So when we talk about diseases, the gene expression is whether or not the gene is active, turned on, and doing what it's supposed to do. So, your genes that code for the proteins that help you grow really kick in at certain points along your lifespan, and particularly when you get to puberty, all kinds of things start happening. Those genes start to become expressed or turned on. You hit adulthood and those genes shut off, and hopefully they stay off because you're not supposed to keep growing, and if you do, that can cause all sorts of abnormalities because not everything grows equally. And that's more of a hormone disorder we'll talk about later. Gene mutations can occur spontaneously, or they can occur due to environmental agents or some sort of outside stimuli. They can occur in germ cells. Germ cells would be our developing cells, so it can be congenital problems, or it can be in the adult cells, the somatic cells. Just a slight word here on gene therapy and what we can do with genes. The human genome, I already mentioned, is about 20 to 40,000 genes. Lots of arguments about where that's really at, but most of the numbers are sitting within that range. We have means of working with DNA now and what we call recombinant DNA technology. It's particularly important with pharmaceuticals where things can be rearranged within the DNA. We talk about genetically modified foods and things, that's a little different, but the same kind of idea. We can now, with technology, combine DNA molecules that are not naturally found together, and it allows for all sorts of new developments and changes in things. Some things can be incredibly beneficial. Gene therapy is what we're looking at if we've got some kind of genetic mutation that we want to fix. There's 4 basic ways of gene therapy, and that's still, even with all of the advancements, I've been teaching this for several years, for almost 10 years now. This is still the same kinds of things, and we're not really very good at gene therapy yet. The idea would be, though, if we have an abnormal gene, we've got a problem, a mutation, to try inserting a normal gene from some donor source. By the way, we do that. I should say use of vectors, not for or vectors. I'm sorry about that. We use use of vectors is how we get that in, and the vectors are often viruses. They've been denatured so they don't cause disease, but they have a great mechanism for getting inside a cell. And they can get inside the nucleus, and we can manipulate them to make things happen. So we can insert a gene, and hopefully the normal gene is what starts copying the right protein, and we're good to go. That doesn't seem to be terribly successful because the normal gene that we put in is not our own, and our cells have a way of saying that's not me and getting rid of it and destroying it. We can remove the bad gene and put in the normal one and try for that one. That too seems like a great idea, but has not been particularly successful. We can repair the gene through processes of select reverse mutation. There's been some progress there. And we can alter gene regulation through the altering of the introns and mechanisms to get things going and get started with them. So it depends upon whether we have the means to turn the gene on and off, so to speak. That one seems to be a little more possible as well. So if you want to read more about it, you can link here. This is the extent of what I need you to know for the course. I just want you to start thinking about some of the genetic therapies that are out there. All right, so looking at inheritance or hereditary diseases, there's some more terminology here. So genotype is an individual's collection of genes. It's the actual codes, the actual genetic makeup. The phenotype is what you actually see that's related to that particular trait or that particular disease. So if you have a certain genetic makeup, we expect a particular phenotype, what it will show and what you will see. The expressivity refers to the manner in which the gene is expressed. So when we talk expressivity, we're using terms like mild or severe or somewhere in between there. So expressivity is the manner in which the gene is expressed in the phenotype. So you've got a gene that's got a mutation. How well is it expressed? Well, it's mildly expressed. It's severely expressed. Sometimes like something like Down syndrome, you might hear someone say they're mildly Downs or they're severely Downs. So you can have the same type of mutation, same genetic makeup, but it isn't expressing as severe. And then penetrance is similar but not the same. It's the ability of a gene to express its function, and it's always represented by a percentage. So expressivity is mild to severe, and penetrance is a percentage. So for instance, if we say a particular gene or mutation of a gene has a 75% penetrance, then it means 75% of the people with that particular genotype will present with a recognizable phenotype. So in other words, if you have, let's say, a gene for blue eyes, and you have the gene for blue eyes from one parent and brown eyes from the other parent. Well, what's the more dominant one, more likely to penetrate? It may be that only 50% of the people who have that genetic makeup will end up with blue eyes, or it's actually much smaller than that, but that's the penetrance of it. So you have that particular makeup in your genes, but not everyone will express that way. Not everyone will have the same penetrance. So you may have slightly blue eyes, that may be the expressivity, but the penetrance, the actual percentage that you would actually show that may only be about 50% of the time. Okay? So much of what we'll look at in this section is single gene traits. So there are disorders that are linked to just one gene problem. But there are numerous, many more diseases that are multifactorial inheritance. In other words, we know that there is more than one gene involved, and there may be environmental factors as well. An allele is a term we use a lot in genetics. It refers to a single copy of the gene. So you have two alleles or two copies of a particular gene. You get one from mom and one from dad. So those are the alleles. Autosomal dominant disorders are disorders in which you need only one mutation on the pair, and we're going to look at that in just a second, in order to have the mutation. In autosomal recessive, you need two copies of the damaged gene in order for that disease to show. So phenotype in an autosomal dominant is just, or I should say genotype in an autosomal dominant is just one mutated gene is all you need. You can have two, but you only need one, and then that your phenotype will show the disorder. How it expresses or penetrance will vary, but you're going to have it. In autosomal recessive disorders, you need two copies of the gene in your genotype in order for it to show up in your phenotype. And then there are some things that are linked just to the X chromosome. So we'll look at those in just a second. All right, so here's your single gene disorders. Autosomal dominant, and here's our picture here showing our autosomal dominant. So the capital D here is the mutated gene. This is normal. All right, so in autosomal dominant, you only needed one copy. Dad's affected here. Mom's normal because she has two normal genes. But in their children, and you don't have to map these out, and we won't do any chi-squares or anything like that. But my point is that you only need one copy of that bad gene in order to have the disorder of whatever it is. So here's 4 conditions that are genetic disorders that I want you to be aware of. Now, I won't get into tons of detail. I just want you to know the basics of each of these disorders. So for instance, Marfan syndrome, an autosomal dominant, meaning only one mutation and you have this disorder, is a connective tissue disorder. It affects the skeletal system, the eyes, the cardiovascular system, particularly the heart, and it often leads to early adulthood death. So we'll talk more about Marfan syndrome in the cardiovascular chapter. Huntington's disease is a progressive degeneration of the cerebral cortex and the basal ganglia. It manifests with what we sometimes refer to as chorea. Chorea is spelled C-H-O-R-E-A, chorea, which is really random, brief, but very abrupt, jerky movements that involves whole body, limbs, torsos, and even the eyes jerking around. That's Huntington's disease. It is a progressive disease that eventually affects the intellectual functions of memory and judgment. What it actually does in the mutation is there's an imbalance of neurotransmitters, particularly leading to excessive dopamine. So that's Huntington's disease. Polycystic kidney disease. The kidneys enlarge and they fill with cysts. The number of nephrons diminish, and eventually the kidney is no longer functional. This disease does not generally manifest itself until about the 4th or 5th decade of life. So not until you're in your 40s or 50s may you ever even know you have it. Huntington's disease also does not usually affect the very young, but shows up usually in early 20s or 30s, and it's a particularly bad condition, but it doesn't happen in youth. It's usually as you get a little bit older. It can sometimes be 40s or 50s, but it can show up earlier. And the neurofibromatosis is a formation of tumors which originate primarily from Schwann cells. They make the, I hope you remember what a Schwann cell does, that makes the myelin sheaths that coat the neuron, the axons of the neurons, or it can also come from other microglia. There are a couple different types of neurophromatosis. Type 1 is the more common, forms skin nodules, and you can see some skeletal deformity with that. May cause kind of dark spots in the skin. And then type 2 is more rare, but it can cause tumor growths. They're not cancerous tumors, but they're tumor growths that can be within the cranium or in the brain and along the spinal cord. So Neurofibromatosis is involved with neuronal tissue. It's autosomal dominant. Then your autosomal recessive disorders. In this case, you have to have both parents passing it along. Because remember, each parent only provides one copy of the gene, so in order for someone to get it, they have to have both copies. In autosomal recessive disorders, you can have carriers. There's no such thing as a carrier in an autosomal dominant disorder because you either have it or you don't. But in an autosomal recessive, in our picture here, these two both have a mutated copy of the gene, but they're carriers. They don't have the disorder. They just can pass it along. And so if two carriers mate, they have a chance of producing a child that's affected. Right? So sickle cell anemia is our most commonly known, and then cystic fibrosis is pretty big in there too, but sickle cell anemia, we see a lot at St. Jude. A lot of patients being treated there for sickle cell from all over the world come for that treatment. That's where the red blood cell is disformed because of problems with the hemoglobin because of this mutation. So they have a problem with carrying oxygen, and it also can be very painful for those oddly shaped or sickle-shaped red blood cells moving through the tiny capillaries. Let me quick do cystic fibrosis, so I just mentioned it. Again, all these are autosomal recessive. Cystic fibrosis is involved in a mutation of chloride channels. It causes an increase in mucus production that then obstructs. It also can affect airways, intestines, reproductive tract, pancreas, liver, sweat glands. All of those areas can be affected. It actually accumulates and affects the respiratory system most profoundly, and so when we get to respiratory system, we'll talk more about cystic fibrosis. Incidentally, I didn't mention this, but sickle cell anemia is predominantly found in those of African descent. Cystic fibrosis, however, is very rare in those of African descent. Then we have Tay-Sachs. Tay-Sachs is a lipid storage disorder. It's due to the lack of an enzyme which results in accumulation in nerve cells. I think I mentioned the Tay-Sachs already when we were looking at cellular adaptations and injuries. It causes lipid accumulation as well in the cells. It affects the brain and nerve cells. It's 100% fatal by age 5. There's maybe a little variation in that age, but no one gets much past 6 or 7 with Tay-Sachs. Even with low penetrance and low expressivity, this is a particularly nasty disease. And then PKU, or phenylketonuria, is a metabolic disorder. It's an elevated essential amino acid phenylalanine. It's caused by a deficiency of an enzyme in the liver that normally converts phenylalanine to tyrosine. And when that's missing, that can't be broken down. It can lead to brain damage, which leads to mental retardations and seizures. However, it's very treatable if you identify it, and we test all infants when they're born for PKU. And you simply modify the diet, reduce meats and poultries, and it's a lot of milk products. So it's a difficult diet, particularly for an infant, but a person can survive PKU. So those are our single gene disorders. There's also X-linked disorders, hemophilia and a form of muscular dystrophy, Duchenne dystrophy, are X-linked. They are linked to the X chromosome. So in this case, remember, females are XX and dads are XY. So dad cannot be a carrier. He has only one X chromosome, and a Y chromosome does not cover for an X because it's different. So if there's an X-linked problem, like our affected male here, he's got it on the X, he's got to have the disease. In this case, if we were talking about hemophilia, he would have it. Now, it's possible for a female to become a carrier, but it's also possible for, say, this affected male to reproduce with a carrier mother. Carrier mothers, it depends upon the disorder. If we're looking at hemophilia, women with a bleeding disorder really cannot reproduce. They cannot give birth. The blood loss of trying to give birth, and even the menstrual cycle in general, can be more than what a female can survive if she has hemophilia. So if she were to get it. So females are predominantly carriers. They usually have a good X to cover it up. Theoretically, she could get an affected dad giving her a mutated X and a carrier mom giving her a mutated X. But in general, moms are carriers. Dads either have it or they don't. And it doesn't really matter which of the disorders. That's the case for something that's X-linked. Multifactorial, multiple things involved. Cleft lip, cleft palate, they're just listed here. These are things that we know have some genetic link. We know that there's some inherited link, in other words, but we don't know the specifics of how they interact, and we certainly see those who have the gene who don't have the disorder, so there's a lot of variance in there. And then there are some that are linked specifically to chromosomes. I mentioned Down syndrome, and there are disorders of XX and XY where there's an additional X or there's an additional Y. So there's some things like that. And then there are disorders that are mitochondrial in nature. So there can be something that affects just the genes involved in the mitochondria. There is specific DNA linked to the mitochondria. These tend to be metabolic disorders. They affect the neuromuscular system. So we don't know much more. We do know more about it, but we're not covering more about it in class. Okay? So that takes us to the end of this. I know I'm looking at a blank screen while I'm talking to you, but let me just quickly say that with all of those different disorders that we looked at, and we talked about parents passing them along and so forth, things can also occur spontaneously. You can have a child get hemophilia whose both parents are completely normal, and the spontaneous mutation occurred, and now the boy has hemophilia, or any one of the other diseases. So you can have Huntington's disease where both parents were totally normal and nothing was passed along, but a mutation occurred. So not everything's as cut and dry as it seems. All right, see if I can shut this off.